Assalamu alaikum everyone and welcome back to Science Tutorials. Today we're diving into topic 12, experimental techniques. This is officially topic 12. I'm covering it as our second topic because understanding the tools that we're gonna use is fundamental before we move on to more complex topics. In this video, I will cover lab equipment, lab safety, simples, and the separation methods will be covered in another video. This topic is highly involved in paper 6. For that reasons, let's make sure that you are familiar with the key pieces of equipment. First, let's look at measuring liquids. All of these tools are used to measure the volume of liquids. And as we know that the volume is the amount of the space that is taken by an object, and its unit is cubic centimeter. Let's start by the burette. The burette is the most accurate tool, and it is used to measure specific volumes of liquids, especially in titration. And also we have tab here. This tab, it controls the amount of the liquid that flows out of the burette. And also we have grading. It starts from the top by 0 cubic centimeter and ends at the bottom by 50 cubic centimeter, and this is the maximum volume that can be held in the burette. And also the burette, you can use it if you want to measure successive measurements. And we will discuss this in titration. The second tool is a graduated pipette. As you can see here also, it's designed to measure small volumes and especially fractions. So if you have small fractions like 3.2, 5.4, all of these fractions are suitable to be measured by graduated pipette. And we can use it by holding your finger up here. So once you remove your finger, the liquid will flow out so that you can look at the grading and you can control the amount of the liquid that will flow out of the graduated pipette. The third tool is a pipette. The pipette, as you can see here, has no grading. And it is used to measure set volumes. Like for example, if you want to measure 5 cubic centimeters, 10 cubic centimeters, you will find a mark here mentions the volume of this pipette. So it measures multiples of 5 cubic centimeters. The fourth tool is a measuring cylinder, and it's very good for measuring large liquids very quickly, but it is not accurate like burette or the pipette. So as you can see also, it has grading, but actually it gives rough measurement. And this is why, because as we know that we measure from the meniscus, and sometimes if you can see properly and you measured from the top of the meniscus, all of this area, I will try to make it with another color, all of this area will be error. So the percentage of the error is high. So that's why it gives rough measurement. And rough measurements, it means not accurate. Here is an example of paper 6. These type of questions are common, so practice labeling to get more comfortable with these types of questions. As you can see here, volume of acid, and he said titration. So during titration we said we use a burette. So the answer here is the burette. He've also titrated, so if you can notice, commonly it is mentioned by titration, and always we have acid and alkali. So which piece of apparatus is used to add hydrochloric acid? It's the burette. A student measures 25 cubic centimeter of an acid accurately. So if he said rough, we will go directly to the measuring cylinder. So here, sure, the burette. So he can change the same question by changing accurate by rough. If he changed accurate by rough, it means this is a measuring cylinder. Moving on to tools for handling gases. Syringe. It is used to collect and measure gases but accurately. So for example, if we hold this in a conical flask and a stopper here, and if there is a chemical reaction here and the product is a gas, this gas will diffuse and it pushes the syringe back. So we can see here this grading, it is measured in cubic centimeters, so we can get the accurate measurement of the gas. The second piece we have here is the gas jar, and this is highly important to know that no measurements, so we cannot measure the amount of the gases by the gas jar. It's just to collect the gases and to store the gases. And here also, guys, there is a problem. Sometimes I brought the measuring cylinder here because sometimes you have confusion between the measuring cylinder and the gas jar. In some experiments, we use the measuring cylinder, but actually you have to differentiate between them by the grading. The measuring cylinder here has grading, while the gas jar has no grading. And here what I'm talking about. As you can see here, this is a chemical reaction and the product is a gas. Why? Because we can see the bubbles. And some students, they label B as a gas jar, but actually it's a graduated or a measuring cylinder. Why, mister? Because you can see here the grading. Here another example. Here no grading, it means it's inverted gas jar. And as I told you here, for example, if there is a chemical reaction and the product is a gas, it pushes, so you can see the measurement of the gas in cubic centimeter, and sure, this is syringe. Let's look at heating equipment. Start with Bunsen burner. It's the source of heat or the flame in our experiments, and usually we draw an arrow below the beaker, so we mention now we're using Bunsen burner. Here you have two holes, one hole for the fuel and the other one here for the oxygen or the air. In one exam they asked what would happen if we close this hole. It means the air will not flow, so there is no oxygen, so the flame will be off. Then the tripod and gauze, they work together to create a stable setup for heating substances. And as you can see here, the tripod, it's a stand with 3 legs. And here this is the mesh, or we can say the gauze. It's like the grill, as you know that the grill, it provides an even distribution for the heat. So same here, it provides or distribute the heat evenly in the container. And when we draw them, we will draw the tripod with 2 legs, and then we add the gauze above here to distribute the heat. And we mention the Bunsen burner as an arrow and write heat down, and then you can place your beaker. So now the beaker is supported by the tripod and the heat is distributed evenly by the gauze. Now sub-equipment used in separation techniques. I will quickly go through each one, but actually I will explain it more in each technique when we talk about separation methods. Here, number 1, fractionating column. It is used in fractional distillation when we separate the two substances or the mixtures according to the boiling point. And then the condenser. Condenser, it cools vapor back into liquid. And this is the shape of the condenser. So we put the water in from here, and the other substance will be moving in this loop inside, and the water will be around it. So water in against the gravity and water out to the sink. So here the function of the condenser to change it into liquid, from vapor to liquid, so it causes condensation. Then the separating funnel, it is used to separate two immiscible liquids, like for example oil and water. So we have a tap here, so we control the amount of the liquid. They will be, one will be up, the other one will be down according to the density. So we can use this tap to take one of these liquids out and keep the other one in the separating funnel. Finally, the filter funnel. If you have a solid, a mixture of solid and liquid, and the solid is insoluble in water, so we use a filter paper, so the liquid will pass through the funnel, which is called filtrate, and the residue or the rest, which is the solid, will stay in the filter paper, and we use a filter funnel in this process. Another question from paper 6, let's label this fractionating column. and this is a tripod. And then he asked to label arrows on the condenser, water in, and here water out. And identify one mistake in this diagram. As we can see here, this is heating to cause the evaporation, so we can condense the gas and collect the droplets here. So this is the collecting beaker. So we can see here an arrow, which means we are heating again to evaporate. So this is a mistake. So we can write heat is below the collecting beaker. Let's move on to the evaporating dish and watch glass. From its name, evaporating dish, so it is used for evaporation, which is a process to change liquid into gas. And it has a large surface area, so it's very helpful in the evaporation. And also it's suitable for heat, so sometimes you will find an arrow and blow it heat, so it's suitable for heat. Come to the watch glass. The watch glass, it is used to reduce the evaporation. Like for example, if you have a beaker, and this beaker it's on a tripod in this way, and here the gauze, and this evaporation takes place, so you can cover this beaker by the watch glass. So in this case you will reduce the evaporation process. And also it is used in another technique or another way. It is used to transfer, for example, the chemicals from the preparation room to the lab. So we can say here used to hold small amounts of solids and drying and cover the beakers. So it is not also suitable to be used for heat. Then the Tissot funnel and the top funnel, both has the same function, are used to add a certain chemical slowly. Like for example, if we have an acid and this acid is going to react with a metal, in this reaction we have to add the acid drop by drop to avoid any aggressive reaction. So in this case we have to use one of them. But the tap funnel, it has a valve or it has a tap. This tap, it gives you more control of the flow of the liquid, so you can control the amount of the liquid according to this tap. Move on to another tools in the lab, like the crucible, for example. As you can see here, it's a small dish. This small dish, we can do chemical reactions that if the chemical reaction requires high temperature, so we can use this crucible. And also we have another thing, this is if you have reasonable amount, but if you have a small amount, we can use the combustion spoon, and it looks like this. And for example, you have here something to protect your hand. We can hold it from here. This is your hand, you hold it and you add the solid that you want to burn here or to heat, and this part, it's like a cover to protect your hand from heating. This is if you have a small amount, you can use a combustion spoon. If you have reasonable amount, you can use the crucible. What about mortar and pestle? You have to differentiate between them. Like for example, this is a pestle and this is a mortar, and both are used to grind the solids. And sometimes we use it, for example, to make an indicator from cabbage. Like if you need an indicator to differentiate between the acid and alkali, we use it. But we add sand, and this is a common question, why we add sand: to increase the friction so we can grind it more and more. Here, this is an example from paper 6. Here you have to label, this is the pestle and this is the mortar, and this is a dropper. And here this is a question from paper 6. Name the apparatus that weigh crystals. So we know this is balance. And here in the other side, this is the crucible. And look at this is watch glass. It looks like the evaporating dish, but the evaporating dish sometimes it's more concave, and sometimes we add an arrow to mention that is heating, so it's suitable for heating, and the watch glass is not suitable for heating. Then move on to the containers in the lab. You have the beaker, conical flask, round bottom flask, and flat bottom flask. All of them, they help to mix the chemicals for the chemical reactions. Out here, this is the beaker, and this is the conical flask. It is mainly in titration, and this is the round and the flat bottom flask. They have the same features, and this one, which is round, it provides, even distribution during heating, and this one, it gives the same function as the round bottom flask, but also it gives more stability because it's a flat from the bottom, so it will be more stable when you place this in the gauze. Also, some tools we depend on the lab, like the test tube or the boiling tube. Both are correct, no worries. You can mention boiling or test tube, and they are used for chemical reactions to mix the chemicals together. And then the test tube rack, it is used to hold the multiple test tubes, like for example if you want to make a chemical reaction and then you mix another chemicals, so you can put them next to each other. And then the U-tube shaped or the U-shaped tube, it is used to purify the gas. Like for example, we put here a liquid, and then the gas will pass from this side, so the liquid will purify this gas and the gas will go from the other side. And here the test tube brush, it is used to clean up the test tubes. Here this is example from paper 6. W is a funnel, so you can write a filter funnel, and X is a boiling tube or a test tube. What about the spatula and the glass rod? Because some students, they have confusion between both. For example, a spatula is used to transfer a small amount of solid. For example, in a chemical reaction, like if you need some solids to be transferred, you can add it here and then you put it in the container like the beaker or any flask. So it is made up of metal, while the glass rod is made up of glass. And why glass? Because it's non-reactive, or it does not interfere in the chemical reaction. Let's move to the test tube holder and the tongues. The test tube holder, it helps to hold the test tube in this way. We put the test tube here, and for example, if you want to heat, it will be much easier for you to heat by using the holder. And the tongues, it's different. For example, if you have a magnesium strips, so you can hold it from this way. If you have magnesium strips, so it's easier to be held by the tongues. And also if you have a hot glassware, like if you have a hot beaker or something, you can hold it by using these tongues. So this is question from paper 6. Let's say together. A, complete the box to identify what is this. This is glass rod because it is involved here in the beaker, so it means it does not react, or it's non-reactive. And then the other question, what is used to add strontium carbonate to acid? So to add a solid, so we need what? We need spatula, so we use spatula. Let's cover measurements you will need during our study. The gas measurement and the liquid, I think we have covered this in the previous slides about how to use each tool to measure the volume. And here the solid, I think you have covered this in physics. If you have a regular object, we use the mathematical equations, and if you have irregular objects, we use the water displacement, and we say the volume of the solid is equal to V2 minus V1. Let's go for this example. Here, this example, by using the burette, the diagram shows this reading. So complete the table. The final burette reading is here, it stops at 13. And the initial, as we know that it begins from the top, is 0. And down, or the bottom, is 50 cubic centimeter. So here the final is 13 and the initial is 0, so the difference is 13. So another question here for measurements. Here this is the initial reading and this is the final reading. This point, just to know from where we read the burette. as we said that if we have a burette in this way, and here this is the tap, and now this is the grading. From up it's 0 cubic centimeter, and it's 50 cubic centimeter from down. So the reading starts from up. So when you go down here, the volume increases. So in this case, we can imagine 0 is up and you go down. So now this is 2.1. So for the initial, 2.1 cubic centimeter. For the final as well, from up to down, 41 and then 0.1. So 41.1 cubic centimeter. Then the difference between them, it will be 39 cubic centimeter. 1 mark for the final and the initial, and 1 mark for the difference between them. Then the time measurement. The time measurement is mainly in seconds. So sometimes they bring questions in this way and they ask you to convert it into second. As we can see here, the circle which is inside, this is for minutes, and the outside circle for seconds. Here this is 0, 1. So now we have 1 minute plus, and here we have second, 15, 20, 21, 22, 23, 24. So 24 seconds. Now we know that 1 minute equals 60 seconds. So we say 1 times 60 plus 24 equals 84 seconds. And also if it's 2, like for example, if this arrow is here, it means 2. So we will say 2 times 60 plus 24 equals, and it will be in second, and make your calculations. Then the mass measurement. In the mass measurement, we use gram, and we use a balance or the digital balance because they give accurate measurement of the mass. But here there is a very important point, which is tare. Tare means you ignore or you subtract the mass of the container. Like for example, if I have a beaker which measures 10 grams, and I have a sample which is 20 grams, for example, of salt or sugar. Now when I put this on the balance, the reading will be 30 grams. So if I want to subtract the mass of the beaker, I will place the beaker first, then I will press enter. So when you press enter here, when you tare the value, it means the reading will become 0 again. So this mass of the beaker will not be calculated. So now when you add the salt, if it's 20 grams, it will appear here 20 grams. The last measurement, it's the temperature. As we know that in the thermometer, the measurement from 0 to 100 degrees Celsius. And here we can see, for example, the measurements. Here it's 10, 15, so we can count 16, 17, 18, 19. So here the measurement, it's about 19 degrees Celsius. And here for this part, it's 20, 21, 22, 23, 23 degrees Celsius. So when we subtract the final minus the initial, so the final is 19 minus the initial, which is 23 degrees Celsius. So it will be minus 4 degrees Celsius. So we add here minus 4 degrees Celsius. Now let's touch briefly on chemical hazard symbols. These symbols help to understand the risk which is associated with the certain chemical. For example, if it's corrosive, it means it will burn the skin. So you will find a hand here which is burned by this chemical, like for example sulfuric acid. Or if it's explosive, like it will cause explosion. Here I have just one point about flammable and oxidizing. They look like each other, but the difference here, oxidizing, you can see a circle here and then the flame around it, but the flammable, you can see the flame around it. Flammable means it catches fire, like for example ethanol. But when I say oxidizing, it's a substance that provides oxygen, that help in burning. So it helps in burning. Like for example hydrogen peroxide. The hydrogen peroxide, it provides oxygen, so it's oxidizing. And toxic substance you can find a skull and bones, and this is harmful. So you can memorize these symbols easily. And now go to the lab safety. These rules are not suggestions. Pause the video and try to read this. And also for each experiment we cover in the future, I will explain the precautions as we go through these experiments. Thank you for watching and see you next time. Assalamu alaikum.